Hi, my name is Gautam Bakliwal. My research subject is universalization of plant stress diagnosis using artificial intelligence under the mentorship of Dr. Heidi Laza. Following are the contents of the presentation. The inspiration of this project comes from the factors of sustainability, statistical agricultural growth, and need for automation. Agriculture plays an essential role in sustaining human civilization. From food to textiles, chemicals, medicines, and clean energy, agriculture is well integrated into every aspect of human survival. Adoption of technology, mechanized tools, high-yield crops, enhanced fertilizers, and pesticides have given a meaningful boost to the industry. However, despite an increase in population and advancements in agriculture, recent years have showcased a consistent decline in previously sought agricultural growth. Declining agricultural interest coupled with population growth, urbanization to more lucrative jobs, and climate change is concerning to ensure global food security. The problem requires more extensive automation in the current agricultural methods that ask for minimum human intervention. This project is based on precision agriculture that has empowered computerized vision to substitute human vision-based traditional manual methods for fast, accurate, and early plant stress phenotyping. The scholarship of this research aims to contribute to the existing image processing techniques in plant stress phenotyping with an objective to foster acceptance of precision agriculture by universalizing algorithm irrespective of plant species and environment using a stepwise approach, to enforce collective sustainable measures and improve overall quantity and quality of crop yield with fast and accurate plant stress diagnosis, to recommend sustainable appropriate countermeasures through stress severity analysis with volume calculation of affected areas. The following system is proposed in the research to fulfill our objectives. The system follows initially where the user selects the plant and inputs leaf images. Leaf images in complex background are segmented and stored in the database. Based on the pre-trained disease classification model of the plant, inference regarding the disease, that are number of infections, percent of infected yield, and infection severity of each disease are provided to the user. Following methods are used in the research to achieve our objectives. For data preparation in this study, we begin with 3,651 high-quality images of apple leaves collected during the 2019 growing season at Cornell Agritech with labeled categories of scab, rust, complex, and healthy leaves. Our dataset accurately depicts practical farm conditions. Raw images from apple leaf dataset are initially hand-annotated for segmentation and equally divided into two parts, each containing 1,825 images to be alternatively used for training and testing in segmentation and classification respectively. In our first method, we implement instance segmentation of leaf using a pre-trained mask RCNN model based on ResNet 101 architecture. Standard data augmentation scheme consisting of random horizontal flips, vertical flips, crops, and Gaussian blur is applied to increase the size of dataset and enhance learning different configurations of leaf. We train the model on a batch size of 2 and following training and validation set. Segmented images from previous method are used in the classification of leaf disease. The segmented output of previous method is fed to a pre-trained ImageNet classification model based on ResNet 101 architecture. The trained model classifies an apple leaf into one of the 4 classes. So the input here is classified into either healthy, scab, rust, or complex diseases. By changing the training data to different plant leaves, different models can be also formulated. The proposed research here initially begins with apple leaf dataset. Some examples of the results from our research are as follows. Our first method of segmentation has an average intersection over union score of 94.8% with following segmentation results. Each image is segmented into a primary leaf. The specific approach helps us to understand and count each leaf only once in our inference and understand the state of the field based on average number of detections of each leaf disease. Our classification model predicted the class of disease from segmented leaves with an accuracy of 95.8%. The predictions from each leaf can be combined to understand the percent of infections of each disease over the plant field. Segmentation of infected areas on the leaf can further help us comprehend the severity of infection over the field. For future work, associating the predicted results with carbon and water footprint can give valuable insights into preemptively figuring the measures to ensure most productive outputs. Based on the predictions, appropriate countermeasures can also be recommended to the user in the most sustainable way. In conclusion, precision agriculture has empowered computerized vision to substitute human vision-based traditional manual methods for fast, accurate, and early plant stress phenotyping. Proposed system efficiently solves the limitations of traditional and existing methods that are labor-intensive and fail upon testing to new crops and unaccounted environments. The research successfully scales precise plant stress phenotyping with improved training times and limited data requirements by applying a multiple-staged automatic hybrid model that requires no expertise in the computational field. Proposed system offers advanced inference relating stress severity and overall state of the plant field to improve quality and quantity of crop yield. The research takes a step forward in the direction of fostering acceptance of precision agriculture by universalizing stress calculation irrespective of plant species and environment. At last, I would like to acknowledge Dr. Heidi Laza for her ongoing mentorship, oversight, and comprehensive review. Dr. Cecilia Monclova as the second reader of the thesis. Dr. Kurt Caswell and Dr. Joseph Hodes as thesis directors. Honors College. The funding support was provided by Texas Tech University Undergraduate Research Scholar Program and Department of Plant Sciences at Texas Tech University. Following are the references used in the research. Thank you.